All right, so with our next talk, we're going to move over into the eukaryotic domain, and we'll have Mary Berbee from the University of British Columbia talking about pectinases linking early fungal evolution to the land plant lineage.
Well, thank you for the introduction, and it's a real pleasure to be here in California. I come from Vancouver, and Vancouver, British Columbia is raining most days instead of being beautiful and sunny like this. So what I'd like to talk to you today are some of the things that my lab group are interested in. And as you'll see, we'll get to why it is that pectinases might be important in early fungal evolution, linking fungi to land plants. My group had 3 goals with the community sequencing project that we've had in collaboration with JGI. So one of the goals is to resolve the branching order of some of the earliest fungal lineages. Another goal is through reconciling the branching order and the secreted enzymes of the fungi, we'd like to resolve what some of those very early fungi would have eaten or used for nutrient sources. And then finally, I'd love to know something about the relationships of early diverging fungi and geological time. So I'll start out with the most familiar fungi, and I didn't show a picture of these because these are the fungi that you probably are familiar with. So these are the Dicaria, and the Dicaria are the Ascomycota and Basidiomycota. So mushrooms, that's one of the groups in the Dicaria, and mushrooms were served as a side dish to the risotto over dinner last night. And yeast. Yeast are also members of the Dicaria, so almost everything that you learned in beginning biochemistry about glycolysis was first studied in yeast. And yeast is also delicious in the form of beer and bread. The other groups of fungi that are in the Dicaria include those various molds, which are probably growing in my refrigerator right now in the yogurt that's somewhere in the back. So those are the Dicaria, the fungi that are most familiar to us. But they're only a small part of the total fungal diversity. And what this tree shows is that in addition to the Dicaria, there are a large group of other lineages of fungi about which very little is known. So unlike the model organisms in the Dicaria, about which a lot's known, these are the mystery groups here. In order to know more about what fungi really did for a living before they got to be molds and yeast, then it's necessary to go back in time and look at those other lineages. So the way to go back in time is to pick the fungi that are representative of some of those ancient divergences and look at them in more detail. So what we really want are the organisms which are a bit like the duck-billed platypuses of the fungus world, or maybe the Araucaria of the plant world. We want the organisms that diverged really early, so by comparison with the more recently diverged organisms, we can get a picture of the geological history of fungi. The people who have been involved in this group are shown here in part, and one of the important people was Dr. Satoshi Sekimoto. He worked on generating the DNA and RNA, which he supplied to JGI, so we could get genomes for some of these early diverging fungi. And another person who's important is Dr. Ying Chang. Ying is doing a lot of the analysis of the genomics so that we're able to come to some at least tentative conclusions about relationships of these fungi. So at present, through our work and through the work of other groups, genomes are now available for all of these early diverging fungal groups, all of these down here. Before we started this work, it was entirely unclear which branching order came first in terms of evolutionary history for these groups. And now we're beginning to see a little bit of the picture. I'm going to be talking about 2 groups of fungi in particular. One group of fungi is called the zygoclade, and from the zygoclade, our group was responsible for a couple of genome sequences in collaboration with JGI. And I'm also going to be talking about the Kittrick clade. I'll start out by showing you some pictures of the Kittrick clade, since these organisms may not be ones that you've seen before. So this is a typical chytrid. Chytrids are aquatic fungi, and they're microscopic, so this ball-like structure here is probably about 30 microns or so in diameter. And the organism feeds through these filamentous structures at its base. What happens is that this structure up here is basically for reproduction. That's where the zoospores will form. And these structures down here are the structures that are associated with feeding. The zoospores that will emerge from this body here they look a bit like this if they're stained in blue stuff. They have a single posterior flagellum and a round head, and they're tiny. They're about 3 to 5 microns in diameter. They are wimps as far as what they can do. They can swim, they can find food, but they can't divide, and they also can't absorb food, so they're strictly for dispersal. The presence of these zoospores indicates a lineage of fungi, which is primarily aquatic, and most of the chytrids have them. So another group of chytrids, chytrid fungi, is represented by this one here. This is a fungus which inhabits not fresh water like most chytrids do, but it actually inhabits the rumen of cattle. It produces zoospores, and it also has rhizoids, which you can't see, but this is a bit of cellulosic material here. And the cellulosic material is providing the nutrients for the fungus. So next time you see a cow, think about the fact that in addition to bacteria, she probably has chytrid fungi in her rumen, which are aiding in the breakdown of cellulose. Most chytrids have those little round bodies that produce zoospores, but some chytrids look more like the dicaria, like the filamentous fungi that are growing in one's refrigerator. This is an example of an atypical chytrid which has a hypha, a filamentous body. It also has zoospores, but it pokes and penetrates into plant material and water, and so the hyphae are presumably an adaptation for that. It also produces zoospores. Next, I'm going to turn to some of the fungi that don't have zoospores. These fungi, these early diverging fungi, are hyphal. They produce filamentous bodies. This one here, Comansia, is often found in soil or dung or insects, and if you're familiar with fungi, you know that that basically means we don't know what it really does for a living. It can be found on all these substrates, but what it actually does most of the time is unclear. What is fairly clear is that the hyphae digest dead organic matter, and the spores, instead of being zoospores that disperse by splashing, are produced on the ends of these, or instead of swimming zoospores, the spores are produced at the ends of branches, and they're distributed by splashing water, a terrestrial adaptation. Another important group of fungi from the zygo group are these. So these are rhizophagus or glomus. They're terrestrial, and they actually grow in the soil, but they also grow in the cells of plants. They aid the plants in uptake of mineral nutrients. This is the hypha of the fungus, and you can see that the hypha here, the filamentous hyphae, is highly branched within a plant cell, and that's the actual increased surface area there for uptake of plant nutrients. The most familiar fungi from the general zygo group are the fungi that grow on fruits, such as, for example, strawberries. Strawberries are red. I don't know what's going on with the projector here. But anyway, zygomycetes take advantage of readily available sugar and nitrogen. They absorb it really fast with their filamentous hyphae, and then they produce spores, which are distributed through air. So what we were interested in doing was looking at these organisms and evaluating whether or not we could get a better phylogeny for the chytrids, and the zygos. We collaborated with Joey Spadafora, who's at at Oregon State University, and we looked at 40 different taxa for which there are genomes available, including fungi and outgroups. And we had 28,807 amino acid positions, which is great. And we had 136 different loci for our analysis. We then built a phylogenetic tree with these data. With genome-sized data, we wound up with much more dichotomous branching order resolve for the fungi and outgroups than had been found before. So for example, if you look at this tree, you can see perhaps a lot of hundreds on the nodes, and that's an indication of good support from the data for the branching order. There are some things in this tree that I really believe, and I think they're going to be things that will wind up in the textbooks for those people who look at textbooks on fungi. So for example, the chytrids, the Chytridiomycota, appear as a monophyletic group. And that is good, because this had been in doubt before. And I'll just tell you the names of the other groups so you can appreciate why this is good. So the rumen fungi were in the potential phyllo Neocalamastygomycota. Teach that to students. The fact that we have a monophyletic group of chytrids means that when we're teaching, we can talk about one group of fungi and attach meaningful characters to them, instead of trying to teach students about a very complex group of fungi that we'd probably wind up glossing over. Similarly, among the zygos, One of the nice results from this study, and also from the work of Francis Martin and his group, is that one of the groups of zygomycota, zygo1 here, includes the molds. There's the mold on strawberry. And it also includes Rhizophagus, the mycorrhizal fungus. So zygo1 then tells us that there is a group of fungi which adapted to land, some of which included members that evolved into mycorrhizal organisms, and some of which became proficient at molding. Okay, so that's all good, but there are still aspects of the phylogeny that we can't resolve to my satisfaction anyway. There's the phylogenetic zone of uncertainty in the phylogenetic tree. This is this zone here in the back where you can see relatively short branches and relatively low numbers, 83s and 89s instead of 100. We were interested in testing the support that the data actually give to some of this resolution to see if we believed whether or not the phylogeny was now correctly resolved. One of the things that seemed like it was worth testing was this. Well, the chytrids formed a monophyletic group, the zygos didn't, and instead zygos turned out to be in two groups, zygo1 and zygo2. So in order to test whether or not this really was the case, that these really evolved separately, we proceeded to do the following test. We looked at the individual loci that had information bearing on the relationship among these taxa, and we asked which of these three topologies was supported by each locus in our data set. There's also the possibility that none of them were supported. So we took basically locus number one and we asked, okay, which of these three topologies is supported? And what we expected was that we would find that most of the loci supported weakly or strongly the tree that came out as the main tree, which is this one here, showing zygo1 and dicaria together. But you probably guessed based on this slide what we actually found, right? Yep, sadly, or maybe not so sadly, it turns out that most of the individual loci, 128 loci, did not support any one of our three trees. Instead, they provided no resolution for branching order. So what that tells us is that the individual loci have very low information content in terms that actually bears on this particular question. And that leads me to wonder about what we see when we concatenate all those loci together. Is that actually telling us the truth? So in summary, I think what's going on here is that the sequencing project goals, that resolving the evolutionary branching order, is being met. And we're seeing meaningful larger groups that are coming together, which is great. But the relationships among those early diverging fungi, among the main groups, is still largely unresolved. And the reason is because those groups share too few amino acid substitutions to successfully link them all together. I'm going to come back to that because I think that that's actually important and results in interesting evolutionary insights. Next, I'll talk a bit about what these early fungi might have eaten. So, the Dicaria, the familiar fungi, are mostly involved in digesting plant material. This is an image showing bird's nest fungi. They're little basidiomycetes. They're maybe about 1 centimeter in diameter. And this part that you see here is the fruiting part. The part that you don't see is the part that's hyphal, and that's actually absorbing nutrients from those wood chips there. So Dicaria are really good at breaking down plant material. And we were interested in finding out whether the early diverging fungi also share cellulases and pectinases for attacking cell walls with those early fungi. Cellulose decomposition is a complicated process. It involves many families of carbohydrate active enzymes. And so this first slide here shows you sort of provides an overview of what the complement of carbohydrate active enzymes is in many different kinds of fungi. So these top fungi here are the Dicaria, and these are the families of carbohydrate active enzymes that are related to cellulose breakdown. These Dicaria have many different copies of enzymes in each family. As you go down to the early diverging fungi, in general, there are fewer copies and less diversity, but look at this, way down at the bottom here, the Chytrid gonopodia, that's the one that had the hyphae, has many of the same carbohydrate active enzymes that are found in the Dicaria. One of the interesting groups of carbohydrate active enzymes is what used to be GH61, but which is now called a copper-dependent lytic polysaccharide monooxygenase. Phew. And the reason why these are interesting to me is because they are fungal specific. And so I was interested in taking a look at whether or not the ones that are present in the chytrid are in fact orthologs from the same evolutionary origin as the ones that are present in the Dykeria. This next tree shows a reconciliation of the organismal phylogeny with the gene genealogy. The yellow branches are the organismal phylogeny, so that's the evolutionary history of the organisms. The blue lines represent the gene genealogy, which is enclosed within the organismal phylogeny. What happened here, what you see here, is that these are the Dykeria, and everywhere where there are a bunch of blue lines, there are copies of the GH61 enzymes, which were inherited from an ancestor and passed on to descendants. But you can trace the blue lines back before fungi, and some of the GH61s are present in the chytrids, and some are present in the zygos. To explain this pattern, one would normally assume that the ancestor had the ability to break down cellulose using a GH61 type enzyme, and that many of the fungi, for one reason or another, lost it. Sometimes the reason for losing it makes sense. So, for fungi that are associated with plants as biotrophs that sneak into a plant and absorb sugars actively, those plants usually don't need cellulases because they're just absorbing sugars. So that includes this fungus here, Eucelago, which is a smut, and smut fungi sneak into plants and absorb sugars without the need for secretion of a lot of different enzymes. And you can see that that fungus has lost the GH61s. Turning to pectins, pectins really interested me because they're one of the polysaccharide classes that are really important now. They're found in the middle lamellae of the cell walls of plants. And they're also found, to a lesser extent, in the closest algal relatives to the plants, to the streptophyte algae. The streptophyte algae are fairly obscure, I suppose, but this is an image of one of them. So Nitella is a streptophyte alga. It's a branching green structure, aquatic. And so our thinking here is that if the chytrids and the Dykeria share pectinases, then their shared ancestor, presumably, would have been associated with cell walls of plants and would have had the ability to break down those cell walls. Now, pectins are complicated, which from my perspective is great. So they consist of a variety of different kinds of sugars and a variety of different kinds of branching chains of sugars. And each different kind of sugar requires a different enzyme to break down the chain. So let's take a look at a comparison between the suite of enzymes present in Dykeria and the suite of enzymes for pectin decomposition, which are present in the chytrid Gonopodia. So this is the chytrid suite of enzymes. This is the Ascomycota Dykeria suite of enzymes. This is the list of enzyme classes, and this is the list of substrates. And look at that. There's almost a one-to-one match between the presence of an enzyme in an ascomycete in Dykeria and the presence of the enzyme in the chytrid in Gonopodia. So that suggests then that these organisms had a common ancestor which was indeed able to metabolize the cell walls of plants. We thought we'd check and make sure that the chytrid really could digest pectin. And so this is one of Ying's experiments. We need to do a few more of these. But this shows the chytrid growing with pectin. This shows the chytrid going without pectin. And the chytrid made much thicker mycelium as well as more growth in the presence of the pectin. So the chytrid really could use pectin as a source of nutrients. So for secreted enzymes, not just Dykeria, but also chytrids have cellulases and pectinases, suggesting that they evolved in association with green plants. The ancestor to the zygos seems to have lost cellulases for some reason. And it also, although I didn't show you the data, lost pectinases. So most of that was somewhat factual. Now I'm going to get into the realm of speculation and try to relate early fungal evolution to geological time. And I'm going to introduce to you the microbial slime hypothesis for fungal origin. You'll see it in textbooks shortly afterwards. So what I'm proposing here is that fungi may have diversified in the Precambrian in oxygen oases where they were digesting algae, animals, and animal exudates that developed in microbial slime. So the most basic thing about fungal biology is this. Plants photosynthesize, animals eat things that are smaller than they are, and fungi hang on to their nutrient source, which has to be larger than they are, and they digest it. So this is a fungus. This stuff around the outside is supposed to represent food. And what fungi do is they need to be inside or on top of their food, secreting enzymes. The secreted enzymes break down the food around their periphery, and then they absorb the food. They don't have any options in terms of getting up and going to some other place. They have to stay right there and complete their reproductive cycle on that food source, otherwise they're dead. So that means that fungi could not have evolved in their glorious present diversity until there was food that was large enough. So what you're probably thinking now limited early fungal diversification. Well, this is actually a fact that oxygen was not at present levels for a long period of Earth's evolutionary history. So Earth's oxygen started to build up with the evolution of cyanobacteria about 2 billion years ago, but it was a long time until oxygen levels reached anything like what's necessary to support multicellular life. This is a chart showing what geologists think happened with Earth's oxygen levels. So this is a logarithmic scale of oxygen as percent of present, and oxygen started to build up about 2 billion years ago. But then it increased rapidly around 700 million years ago, which correlates with the time when or it's shortly thereafter in terms of, well, geological time, I guess it's millions of years thereafter, complex animals started to appear in the fossil record. And a couple million years after that, land plants diversified. The reason why complex animals could not have diversified much before that was because of the diffusion limits of oxygen moving through water. So oxygen, first of all, had to get to the animal, and then if the animal was multicellular, oxygen had to diffuse from one cell to another, which is not an easy thing to accomplish at a low oxygen potential. So if you imagine a billion years ago, there were the unicellular ancestors of both animals and fungi. And it's vaguely possible that fungal diversification paralleled the Cambrian explosion of animals. So this is about 650 to 541 million years ago. And that's the time when animals, worms, mollusks, and all of the major lineages of animals started to emerge. Well, perhaps that was also the time when all of the major lineages of terrestrial and aquatic fungi started to diversify. Remember, I told you that in the fungal phylogeny, there's the zone of uncertainty. Well, that zone of uncertainty might be telling us something true about the evolution of fungi. It could be that the ancestor to all these taxa was present 600 million years ago, but then it required the oxygen levels to increase before the lineages that are now familiar diversified. And it could be the period of diversification was very short relative to the amount of time that came afterwards. So what nutrient sources were available during the time when fungi might have been diversifying? What would have given the fungi enough food in order to reproduce? Well, land certainly wasn't going to do it. So this next image is from geologists. And it shows land in the early Cambrian or late Precambrian. This represents mountain ranges that were forming. And this represents streams, all braided streams. They weren't forming proper V-shaped stream channels, and the reason for that is believed to be because there were no land plants with roots that were changing the channel shape. So there was a lot of shallow water here, and then there were the shallow seas around the outside. Where were the organisms at this time? Well, there was multicellular life. And there's geological evidence that the multicellular life consisted of things that lived in the shallow seas, possibly also in the shallow freshwater. And these animals This is an animal, a slug-like thing, and those are clam-like things. Those animals lived in an area of microbial slime right at the interface between the anoxic soil down, or anoxic sludge down below, and the hypoxic water up above. So what did microbial slime look like? Well, some of the geologists recreated microbial slime in a laboratory. They thought it might look a bit like this. So there you can see the microbial slime has a plant layer on top. This is the surface view. And then in cross-section, the microbial slime consisted of a layer of algae mixed, and perhaps in reality, algae mixed up with a variety of other kinds of things. So 600 million years ago, maybe it was this layer of microbial slime here, which supported the early fungal life. That microbial slime might have included the streptophyte plants that eventually gave rise to the land plants. And it certainly included animals and animal exudates, which might have served as food for the early zygo lineage, which you may recall lack most of the cellulase enzymes. So next time you see a story about Burgess Shale or about early life in the oceans, think to yourselves, fungi were there. Where were they, and what were they doing? And with that, I'd like to say thank you so much for inviting me, and I'd also like to thank the AFTAL II U.S. National Science Foundation for funding, and National Science and Research Foundation of Canada, as well as the various people who have given me permission to analyze their genomic data. Also, thanks to my students who are always willing to listen to my practice talks and provide useful criticism. Thank you.
I think we have time for a couple questions. Look like Igor. Can someone get a mic to him so that everyone can hear the questions? Oh, looks like we've got one coming from that side.
Thank you. Mary, you've shown that there is a match between enzymes in Kittrick and Dyke, but one of the examples that you mentioned is Pyromyces with significant expansion. Can you comment on that?
That's absolutely fascinating. So Pyromyces is a Scott Baker sequenced organism in association with JGI. And so really, Scott should comment. Okay, in the absence of Scott, would you like me to comment? Okay. So I think that Pyromyces must have inherited the same suite of enzymes that are found in the other chytrid, the Gonopodia. But it doesn't have many of those enzymes now. It looks like what happened is the Pyromyces hijacked enzymes from bacteria that were also present in the guts. So the enzymes of Pyromyces, in most cases, appear to be of recent bacterial origin. And they're bizarre. So instead of having a single carbohydrate binding domain, they've got 6 of them or something like that. And then there's the actual catalytic domain. So they really went crazy within the rumen of the cows.
Do you have an idea of the dates on the diversification of the early fungal groups? Is the tree dated?
The tree is not dated, but I'm of course thinking about that. So I think that you can make a maximum age for fungi based on the oxygen level, for that diversification based on oxygen levels. So fungi predated that oxygen spike at about 600 million years ago because they share an ancestry with us, and that's estimated to be about 1 billion years ago. But I think about 600 million years might be a maximum age for that diversification.
All right, well, let's all give Mary another round of applause.